Hey guys, welcome back. Every day I am uploading a video or taking a live session regarding class 10 biology syllabus in order to complete the syllabus in right time. If you are a class 10 student and want to learn through my sessions, then subscribe and press the bell icon so that you would not miss any notification regarding my live sessions and the videos that I upload. Today's topic is plant tissue culture from the chapter How Do Organisms Reproduce from class 10 biology. Here, tissue culture was learned by scientists during 1950s. I repeat this once again. Tissue culture was learnt by scientists during 1950s, and in this tissue culture method new plants are grown by using living tissues or cells of the plant called explant. So this is one of the very important word in tissue culture. Explant could be any part of a plant which is used for culturing. I repeat this once again. Explant could be any part of the plant which is used for culturing. Okay? Now here the cells are placed in an artificial nutrient medium where they divide rapidly to form callus, and here the callus is undifferentiated mass of cells. I repeat this once again. Callus is undifferentiated mass of cells. If you look here in this picture, see, an explant is taken from this plant, and here the explant is kept in an artificial medium which is placed inside a petri dish or a test tube. You can see here, okay, sterilization and growth on suitable medium. Sterilization is performed so that the harmful microbes which are present on the explant surface could be killed. Then they are transferred into the suitable synthetic nutrient medium. Like here, synthetic medium is present in a petri dish. Now inside the explant is kept. Now what happens? The callus is formed. This is nothing but the callus. So callus, as I told you, it is undifferentiated mass of cells. It means all the cells in that mass are same type of cells. Okay. Now here this nutrient medium which is used must provide carbon source. Okay. So this nutrient medium must provide a carbon source such as sucrose. This is one example, and it should also contain inorganic salts, and also vitamins, amino acids, growth regulators, plant growth regulators. Okay, so this is like auxins, cytokinins, etc. So all these must be present in this suitable nutrient medium. I repeat this once again. The nutrient medium should contain carbon source like sucrose. It should contain inorganic salts, vitamins, amino acids, plant growth regulators like auxins, cytokinins, etc. So in presence of all these favorable conditions, the plant cells or tissues, they grow as undifferentiated mass of cells. That is what we call callus. Okay, so here this callus is now transferred to another medium containing nutrients and hormones for growth and differentiation. So here a fresh nutrient medium is utilized in the second time. Now if you look here, this is a first used nutrient medium. Now in these test tubes, the nutrient medium which is present is a fresh nutrient medium which has along with the nutrients the plant growth regulators which are needed for growth and differentiation process. So here the callus is transferred into a fresh nutrient medium that contains nutrients as well as plant growth hormones. So in presence of these plant growth hormones, these callus will grow, it will differentiate, new roots and shoots are formed in the plant parts like this. You can see here the root formation and also the shoot formation. This is because of the plant growth regulators which are present in these nutrient medium. Okay, now moving to the continuation, the plantlets are formed and they are placed in the soil so that they can grow into mature plants. You can see here, once the plantlets are formed here, they are transferred into the soil, and this is how finally we get a matured plant, and this is the technique we call plant tissue culture. So here within short span of time, large number of plants can be produced, and here the parts of the plant like explants can be cells or tissues, and by using these cells or tissues a whole plant can be produced. And this ability, one of the very important word, this ability is called totipotency. So the ability of the cells to form a whole plant, the ability of a tissue to form whole plant, this ability is called totipotency. So this is the reason behind the growth of new plants from the plant cells and tissues. Now let's discuss about advantages of plant tissue culture. Here large number of plants can be produced in relatively short span of time and a small space. And here this method of producing thousands of plants through tissue culture is called micro propagation. Okay, so here I repeat this once again, a large number of plants, thousands of plants can be produced in relatively short span of time and small space by using tissue culture. And this is what we call micro propagation. Many important plants like tomato, banana, apple, etc. So these plants have been produced in commercial scale using this method. So I repeat this once again, many important food plants like tomatoes, banana and apple, etc. have been produced in commercial scale by using this method. And one more important advantage here is this technique, the plant tissue culture, helps in crop improvement by producing disease-free plants. So by tissue culture we can produce disease-free plants even if the plant is infected with a virus. Okay, the meristem like both apical and axillary, okay, is free of virus. I repeat this once again. Okay, even if the plant is infected with a virus, the meristem, that is apical meristem as well as the axillary meristem, it is free of virus. Such meristems, hence one can remove the meristem and grow it in vitro conditions to obtain virus free plants. So the meristems are obtained, they are grown in vitro conditions in laboratory, and as a result virus free plants can be produced. This is how disease free plants or the virus free plants can be produced. Scientists have succeeded in culturing the meristems of banana, sugar cane, and potato. Okay, so already scientists have succeeded in culturing the meristems of banana, sugarcane, and potato. So this is how the plant tissue culture helps in crop improvement and by producing disease-free plants.